All right, folks, so we should get started. So we are now at the last session of the workshop. And the theme of the talks that you'll hear today is biology. So we are starting out with Josef.
All right, thank you. And thank you for the organizers for inviting me to give a talk here. I feel like a bit of an outsider because I'm not an expert in AI or machine learning. I am a domain scientist who studies biodiversity science and systematics. But today what I want to talk to you about is how I see potentially knowledge-guided machine learning really accelerating discovery in my field. I consider myself an evolutionary biologist, and evolutionary biology is fundamentally the study of variation. So biodiversity science and evolutionary biology have variation at their core. Variation is the engine of evolution. And if you go to a natural history museum, you will see data that looks like this, specimens preserved, many of them over 100 years old, and just incredible variation of life across these collections. And if you go to these museums as well, you will find people who are experts on these groups, right? These are the domain scientists who are focused on a particular group of organisms which they know very well. This is University of Michigan Herpetological Collection, and they absorbed my PhD advisor's collection. So some of those snakes you see up there, I collected in my PhD, and this gave me a very intimate knowledge of the features of these organisms. So when I say this field of systematics, that's kind of the core of museum science, core of biodiversity research, what is it? So this is a three-pronged science that involves the act of taxonomy. So taking the variation that we see in those drawers and assigning labels to those individuals. So species, describing new species, and the nomenclature that goes with that, assigning labels, assigning individuals into those taxonomic concepts. But we don't just want to label these species, we also want to understand their evolutionary history. And so we want to construct the story of the evolution of these groups through building of phylogeny. These are phylogenetic trees, they are graphs which show us the evolutionary history, and we want to link those graphs to these sorts of events that have happened in Earth's history, in the history of a group. And we also want to just understand the process of evolution itself. Why is there this variability? What are the genetic components of this variability? How do they interact with their environment? Why do some groups evolve a ton? Why do some groups not evolve at all? And kind of the currency of a lot of this work is the study of traits or features of organisms, the visible features of organisms that allow us to do things like discover new species. So even with our genetic data, a vast majority of new species are still discovered based on experts looking at them and seeing the visual features that identify them as novel species. And by the way, we may think that those museum collections are where we deposit new species, voucher specimens that represent those new species. But when you see new species discoveries, a lot of them are not, I discovered a new species in the wild. It's, I discovered new species in a drawer that have been sitting there for 100 years, right? And so as biodiversity is lost, there's a need to have expert eyes on all these specimens in a way that is simply not scalable given the number of people we have that are experts in these groups. But besides describing new species, we also want to use these to help us build phylogeny and infer the evolutionary relationships. This may seem obsolete in the age of genomics, but with we actually need to, in order to build phylogenies that are placed in the context of time, we need to use fossils for which genetic data aren't available. And there's all sorts of reasons that we want to use these visible traits. We also want to link these to the genes that are underlying them, the functions they have for the organism, and then how the environment shapes those traits over time through the process of evolution by natural selection, and understand why some groups diversify a lot and why some groups don't diversify at all. And so the field of phenomics has emerged to facilitate this, which involves taking high throughput data of phenotypic traits from organisms, from images of organisms. For example, you could landmark features of different organisms in this field of geometric morphometrics, image segmentation, or maybe representing the colors of organisms in a way that can be computed and converted into these traits that we can study. And this is getting ever more sophisticated, ever more complete representations of these museum specimens. So this is a frog that's been CT scanned in various ways so that it has incredible resolution of this museum specimen. It has incredibly complete data on the phenotype that can be accessed by anybody in the world. And the hopes is that this will accelerate discovery in biodiversity. But I will argue that while genomics has undergone a data revolution, comparable advances in phenomics have lagged. And the reason for this, I'll walk through here, is that for genomes, we have a very well-defined nucleotide state space that's amenable to automation, ACs, Gs, and Ts. Whereas with phenomes, how many traits does an organism have? How would you tackle that cupboard of insects and start defining traits that are meaningful to measure? There's nearly an infinite number of ways you can take a measurement, but the organism doesn't have an infinite number of traits. This leads to high reproducibility, big data in genomes, but a reliance and a bottleneck in phenomics and biodiversity research for how we are going to come to a consensus on how to represent the variation within these groups. In addition, the process of molecular evolution ensures that these sites in a genome are relatively independent, although more or less, depending on your view. With phenotypic traits, they're incredibly integrated and dependent on each other. What this means is that genomes are amenable to statistical computation, but phenomes have the curse of dimensionality and extreme challenges in representation. So we are already dealing with black box models in biodiversity research. We are dealing with domain expert biologists who we rely on to construct our primary data. And the problem isn't so much that the data isn't available, it's that there's a bottleneck in representing that data in a way that can be used to ask the scientific questions that we are interested in. And so if you looked at that drawer, would you immediately pull out these as the most important categories of variation for these different? It takes a naive scientist approaching this versus somebody who has studied these organisms for many decades will come to different traits that they build, okay? And so this is why biologists value this so much and why they're so freaked out all the time, because there's an increasing risk to these collections as they are moved around and not preserved in ways and valued. And the work itself, this work of going through all these specimens, is not valued as a science either. And so what you get is that these scientists are in a race to make this data available in the hopes that something will come of it. And that largely hasn't happened because the bottleneck is in those experts who can construct meaningful characters that the community can use to study evolution of traits, becoming a lost art. And so what are they doing in their black box to construct these characters in a way that's meaningful for these questions, right? And so my graduate student, Caleb, who helped me with this talk, found these two quotes. And so this one is from David Hull's paper and colleagues' paper, Phenomics, The Next Challenge. And he says, rapid progress on any biological problem rests on the hope that there is at least one viewpoint to each problem that makes causation relatively simple. And I think that this is something that a lot of us I would agree with, at least. And here's another quote often attributed to H.L. Mencken, a reporter and supporter of Teaching Evolution in the Scopes Monkey Trial. For every problem, there is a solution which is clear, simple, and wrong. And these two seem somewhat at odds with each other, right? Because I also agree with this, right? And so what's the difference here? Well, Hull was talking about causation, right? He had the keyword of causation, right? And so something that can really assist phenomics is understanding, or high-dimensional phenomics measures are still prone to redundancy, ascertainment bias, and incompleteness. We don't know how to represent these high-dimensional phenotypes in a way that captures all of the biological meaning we are after. Latent phenotyping methods can address these problems, but they can be difficult to interpret and evaluate. And so I think knowledge-guided machine learning can resolve some of these issues. And of course, you can incorporate knowledge of something like, if you're an experimental biologist, you can incorporate knowledge of the experimental structure into the model's architecture. So here's an example paper that does this, a latent space phenotyping for treatment studies. And the basic idea here is that you have two treatments in an experiment. You have a time series of photos of, in this case, a plant exposed to that treatment, and you train a neural network to distinguish between the treatment and the no treatment treatments, and produce an experimentally structured latent vector that you can then use as a trait to pass on to GWAS studies and the like. And here you don't necessarily need to know the exact features, but you're trying to identify the features that respond to the treatment that are genetically based. Now, as an evolutionary biologist, I don't have access to million-year experiments, right? But ancestry is the critical causal structure relevant to genetics and evolution. So if I have a bunch of individuals that vary, and I have an ancestry graph, so if these are different individuals in a population, they have ancestors, they have parents, they have grandparents, we can have a pedigree that provides us a structure of inheritance that we can then use to partition heritable variation from non-heritable variation. And that heritable variation is interesting to evolutionary biologists because it's what responds to natural selection and evolution. Or if those entities are instead species, we can construct a graph that represents the splitting of lineages over time as they speciate and diversify into the many different species we see today. And this is called a phylogeny. And this can be so when we think about systematists who are looking at images, they have a hypothesis of this relationship in their mind, and they're using it to help them to identify traits that have meaningful phylogenetic signal in the data when they construct their characters. And this is important because if you look at raw images, and so this is a paper from Anuja's group, looking at the saliency of different features of fish for species classification, the model can be correct for very wrong reasons. So here it's picked out parts of the outline, here it's picked out some parts of the eyeball, and here it's read the species label to identify the fish, right? And so that may be good for identifying it's a species, but it's probably not going to be good for identifying to these higher level orders, right? Because the consistent thing that's going to have phylogenetic signal in these images is the fish itself. And so this is part of the premise of this large HDR working group that I'm a part of called Imageomics that Tanya and Anuj are also both part of, that seeks to extract biological traits from images. And one of the projects that I was lucky enough to be a part of that was really led by Anuja's group is discovering novel biological traits using phylogeny-guided neural networks led by Mohamed El-Hamad. And this neural network seeks to embed the phylogeny into the species classification problem by having these multi-labeled images and then assigning these features at different levels of the phylogeny and predict classes as we go up this hierarchy. And so to kind of show how this works, we can take an input image into this phylo encoder, and then it gets split into two vectors, which here we call the imageome. This first one here is a phylogenetic vector. So these are features that predict these phylogenetic features going into the image, and then these features are what's left over. So these could be environmental features, or these could be the features of the organism that evolved too quickly to be observed on the phylogeny. And what we get from this is phylogenetic embedding distances here in this distance matrix heat map that align with the actual evolutionary distances, okay? And so these features are hopefully these kind of evolutionary traits that have phylogenetic signal and be useful for placing other fish into the phylogeny and probably respond to natural selection and evolution over time. If we do species to a species image translation by replacing those features iteratively, we can see that the phylo-NN, as we go from one species of fish to the other, produces a sequence that if you look at those fish, at least with my biologist eyes, they look more fish-like than the non-phylo-NN models, where you have things that don't look very much like fish. And the hope is that these are biologically plausible transformations through evolutionary time. However, is this correct? Is this actually what the ancestor of these things looked like? I have no idea because those fossils don't exist. Our data is incredibly sparse. And so what we are really interested in is, can these models use the information in the phylogeny in a way that pulls out traits that are going to be meaningful for the questions we have? And it's going to be very hard to be generalized across all of these different particular groups unless the only thing that's in common is they all have a phylogeny and they all have traits that are going to align with that phylogeny. So what we are targeting are these plausible transformations in the trait space. And so my graduate student Caleb has been building simulation pipelines. There's well-known GP maps jargon there. There's well-known mathematical models for translating genotypes into the morphology of, for example, snail shells. We can use these to generate data sets where we have, we're not limited by the size of the data. We can generate as many images as we want of species that have evolved. And our target is to infer the underlying traits that are independently evolving in these different species. And we can use this to then validate our phylo-NN tools to see if we are getting using the information as an evolutionary biologist would to construct these traits and construct these characters for downstream tasks. Okay, so running out of time here a little bit, so I'm going to speed through the rest of this. So we're continuing this work, and Anuj is continuing this work with, so Maruto Kurana has this paper that extends the phylo-NN and allows us to essentially take features that are learned in a particular branch of the phylogeny and swap them into another lineage and generate an image of what that fish would look like if the events that happened on that phylogeny had occurred then. This is essentially like what molecular biologists do when they do gene editing, when swapping genes or knockout experiments. And what this can help us understand is how this variation is in these evolutionary phenotypes is partitioned at different points in this phylogeny. We're also trying to link this to vast knowledge bases. I've been part of this project called Phenoscape that uses phenotypic ontologies, the identities of those traits, to link our characters together. And with those, we can then link our characters from the evolutionary phenotypes to model organisms through these ontological knowledge bases. And the hopes being that we can find the genes underlying phenotypic traits. Our hope is, similarly with knowledge-guided machine learning, that we can take evolutionary phenotypic data that is structured by phylogeny, find the independent traits that are evolving on those trees, take model organism data, also find the latent phenotypic space of those genetic manipulations, and then link those together so that we can potentially understand how these transformations are actually mechanistically happening across these scales. So takeaways, latent phenotype methods and KGML can overcome biases and bottleneck. Traditional character construction, knowledge-guided machine learning makes latent discovery more interpretable and meaningful to biologists, and we can validate it with these simulation techniques.
Yeah. Right. Let's thank Josef for the wonderful talk. We have time for questions.
That was a wonderful talk. Thank you so much. I had a couple of questions on the autoencoder architecture that you had there.
Yeah.
And I want to make sure I understand how that works. So you have a loss function. Okay, this one. Yeah.
Yeah.
So you have an image coming in, and there's the autoencoder. Does the autoencoder loss function include both the reconstruction as well as deviation from some reference phylogeny, or is it only reconstruction? In other words, the key question is, do you have a reference phylogeny that you're trying to match?
It's probably best for Anuj to answer. but-
So the question was that we are using a reference phylogeny or not?
Yes, we have a known phylogeny.
We are using a reference
So
phylogeny.
then the next question. So if the goal is to construct the phylogeny itself,
Uh-huh.
if that was the goal,
Yeah.
then basically your autoencoder would have to use as loss function the image reconstruction, as well as some characteristics of what you want the phylogeny to look like. For example, you might want a very long phylogeny or maybe a bushy phylogeny, things like that. How would you encode that into the loss function of the autoencoder?
Right. So I would say that typically in these studies we already have a backbone phylogeny from genetic data that is going to far outpace the information we can gain from the phenotype data, and our task more is placing new images within the backbone of that phylogeny rather than building the phylogeny from scratch.
But might it be an interesting question to ask to reconstruct that phylogeny? Because there are a number of distance assumptions that are made in reference phylogenies, and
Yep.
these things allow non-linearities, which
Sure.
probably are more consistent with biological evolution.
Mhm.
So maybe you want to integrate both the phylogeny construction or reconstruction and image reconstruction into a single loss function here?
And just to add to that, so we are using phylogeny obtained from molecular data. The images that we're using can fool us too, because they can be convergent in evolution. Two species that are very different in phylogeny may look very similar. Okay? So we're not going into the image space for similarity. We are relying on knowledge that we know from molecular data to guide us in our future. Yeah.
So I was just thinking that your slide with actually the same thing, right? The fish sort of transforming from one, your right, exactly this. From an evolutionary biologist's perspective, you are doing what Darcy Thompson tried over 100 years ago. Darcy, and he has this book on growth and development, where he's actually done this sort of thing with sketches, right? So he was also interested in the environment, right? What was driving evolution? Is there the possibility of, now that you're now working with sort of mapping phenotypes for evolution, is there also the opportunity to take another step back and connect that with the sort of driving forces from the environment? You sort of might know what a particular species, what environment a particular species operates in, and
yeah, absolutely. So we have this very sparse data of living species. We have the environment they live in today, and maybe a little bit in the farther back. And so in order to make that inference of how is this environment shaping the trait change in each species, you need to account for the history, the phylogenetic history of all of where they're coming from. Where did they start, right? And so that is absolutely a target that we are after. Yeah.
All right, let's thank Josef again for the talk.